Okay, good morning, good afternoon, and good evening to everyone tuning in. Thank you so much to the organizers for inviting me to present. My name is Margaret Krause, and I'm currently a postdoc with the International Maize and Wheat Improvement Center, or CIMMYT, in Mexico. And today I'll be presenting some work that was actually part of my graduate studies at Cornell University, but it was also done in conjunction with CIMMYT to try to bring in high-throughput phenotyping into the bread wheat breeding program at CIMMYT. So how can high-throughput phenotyping benefit breeding programs? I like to demonstrate this by putting up this aerial photo of the Campo Experimental Norman E. Borlaug in Ciudad Obregón, Mexico. It's the largest breeding station that CIMMYT has in Mexico for wheat, and each year hundreds of thousands of lines are planted both for the breeding program and for research purposes. And this is a photo of me in just one of those plots. So as you can imagine, despite having really excellent staff at CIMMYT, it's just not possible for us to get around to every single plot and do really extensive quantitative phenotyping. So that's what these advances in remote sensing really allow us to start doing. We can do more extensive field-based phenotyping with reduced labor, time, and cost in a way that we weren't able to do before. The other point that I'd like to make is that HTP at this point is no longer new. Over the past 10 years, the community has done a lot of really great work in terms of research and development for these technologies. And what we're seeing is that for the most part, it's working in the sense that the traits that we're recording with these HTP systems are oftentimes tracking with our traits of interest. So we're able to start moving out of the research and development stage and into more of a validation and implementation stage in breeding programs. So that's what we wanted to do within the CIMMYT bread wheat breeding program. This is based off of several generations of selected bulk, and the F5 or F6 is really the first time that plants are selected individually and become derived fixed lines. However, because of limited seed yield per plant, there's insufficient seed to plant first year yield trials right away. So typically these are grown in these small plots for the purpose of seed increase, and these are 2-row 0.7 by 0.8 meter plots. They're unreplicated, they're hand planted, and typically more than 40,000 are planted at the Campo Experimental each year. So measuring grain yield on these plots is typically not feasible. And they're promoted to first year yield trials based on visual selection in the field for agronomic traits and after harvest for seed traits. And so because we're not able to feasibly measure grain yield, but we could theoretically measure high throughput phenotyping traits at this stage, we thought that this could be an effective avenue for introducing high throughput phenotyping into the breeding program. So that was the goal of the project. And so to test this, we planted over 2,000 breeding lines in 2 different ways at the Campo Experimental. One says these unreplicated small 1 by 0.8 meter plots that I'm calling small plots, or SP. And then again, as these twice replicated yield trial plots, or YTs. And these images can show you kind of to scale how these look in the field. We phenotyped both with a Matrice 100 unmanned aerial vehicle, or UAV, by DJI, equipped with a red edge multispectral sensor by MicaSense. And briefly, what the MicaSense does is it records the amount of reflected light off of the crop canopy at different wavelengths in the light spectrum. And with this information, we can calculate vegetation indices such as normalized difference vegetation index, or NDVI, and previous work has showed that NDVI tracks closely with grain yield in wheat. So in addition to NDVI, we also measured grain yield in both the YT and the SP, although the breeding program does not typically measure grain yield in the SP. So we were interested in evaluating NDVI measured in the SP as a potential selection criteria for improving grain yield. For selection criteria to be useful, they must be first heritable and second correlated with the trait of interest, in this case replicated yield trial grain yield. So first, looking at the heritabilities, we saw that the heritability estimates for NDVI were similar across the YT and SP. So despite the difference in plot size and replication, we were seeing similar levels of heritability heritability for NDVI. And we were also seeing considerably higher heritability for NDVI than for grain yield, particularly in the small plots. So we're seeing that these traits are heritable. The next step was to look at the phenotypic correlations. And within the YT and the SP, we were seeing moderate correlations between NDVI and grain yield. They were a little bit higher within the SP than within the YT compared to the SP. Across the YT and SP, we were seeing high correlations for NDVI. So despite the difference between the plot sizes and levels of replication, we were seeing that NDVI was generally tracking the same signal. And then finally, the relationship that we were most interested in was between small plot NDVI and yield trial grain yield. And we were seeing moderate correlations of 0.3 to 0.4, which suggests that if we selected on small plot NDVI, we could potentially see increases in grain yield at the yield trial stage. So our next question was, how does this compare to what the breeders are doing, which is visual selection? So this was a separate experiment in which we took 300 plots from the breeding program's small plot stage. These are a little bit smaller, they're 0.7 by 0.8 meters, and we harvested those for grain yield and we also phenotyped them with a UAV for NDVI. Within these plots, the breeders did visual selection and selected 36 plots at a selection intensity of 12.3%. And so on the right-hand side, on the top, I've plotted NDVI against grain yield for 2 time points, and the solid dots are those that were selected by the breeders. And when calculating the response to selection for grain yield, we calculated a value of 0.38%. For NDVI, the top 36 lines would be those to the right of the dashed lines for each time point. And when calculating response to selection, we had values of 1.54% and 1.4% for the 2 different time points. So this was considerably higher than visual selection. However, we also recorded qualitative scores for days to heading for all of the plots, and that's what the colors in the figure represent. So you can see that the breeders selected sort of a mixture of early, mid, and late lines, whereas due to confounding between days to heading and NDVI in this population, NDVI would have primarily selected late and very late lines, which typically is not desirable because CIMMYT is trying to develop varieties for a wide range of geographic locations. So I thought about different ways that we could correct for this, and I was reflecting on how breeders, when they're in the field, they're considering all kinds of different traits all at once. So I developed an approach that would consider both the NDVI and the qualitative days to heading scores, and within the 36 plots that were selected visually by the breeders, 22, 8, and 6 corresponded to the early, mid, and late groups for days to heading. So what I did was I identified the top 22 lines for NDVI within the early days to heading subgroup. And I did the same thing for the mid and late subgroups and identified the top 36 lines that way. And that's what's represented in the bottom graph. This time the colors represent whether the lines were visually selected by NDVI, or selected by both methods. And the response to selection for this approach was actually even higher at 2.9% and 4.23%. So if we had relied fully on the NDVI values, we would have had that undesired shift in the distribution of phenology. And this other approach highlights how we can leverage useful information from HTP in conjunction with other data that we might collect in the breeding program. Unfortunately, I don't have time to talk about the other things that we did with this project, but I'd like to direct you to an article that was just published in Crop Science on this research. This is open access, so please go check that out. And finally, I'd like to acknowledge my advisors at Cornell, Michael Gore and Mark Sorrells. On this project, we worked very closely with Jesse Poland in the USAID Wheat Innovation Lab at Kansas State University. And then at CIMMYT, I worked within the bread wheat breeding program, and specifically with Suchasmita Mandal. And then Jose Krause of the biometrics unit served as my mentor during my time there. My graduate funding was provided by the U.S. National Science Foundation and the U.S. Borlaug Fellowship in Global Food Security, and the project was funded more broadly by USAID Feed the Future and the Delivering Genetic Gain in Wheat project. So thank you so much for your interest, and I'm eager to interact with everyone in this virtual format. Thanks so much.